Goal of my talk today is to sort of give a little bit of a background of why I'm interested in what I'm doing. Then I will introduce in a very global way how microbial mats produce carbonate minerals, which things are involved in that, and then I will look at two different field sites where I've been working, so to connect the fossil record deep time Archean with some modern analogs perhaps that we have been able to find. So of course, as you know, I'm preaching to the choir here. If you work in deep time, especially as deep as the Archean, you never have more than maybe 2 or 3 or 4 pieces of 1,000 pieces puzzle, right? And so you have to be creative and try to collect evidence to see to make a case, and we will never know exactly what happened to the Archean, but we come closer and closer with newer techniques and what have you, and newer ideas. So since some of you, I have to close this thing. Got it. I have something in the middle of my screen. Can I forward? Yeah, I can forward. Here we go. We go back to 4 billion years ago in the beginning of the Archean, where we see the end of the late heavy bombardment, and maybe some liquid water, some magma rafts floating in this ocean, the moon still very nearby, and Earth is reforming. We think that shortly after this, life may have started, and things quieted down a bit. In the Paleo-Archean, maybe a little bit in the Eoarchean as well, so 3.5 to 3.78 billion years ago. If any of you have been to the Smithsonian in Washington D.C. and you start with the beginning in the museum, you see this backdrop, an artist rendering of what life may have looked like in the very beginning. And of course, what you see here is you see the volcanoes, you see a dimmer sun, you see lots of CO2 in the atmosphere, a very dense atmosphere, and you see these structures at the border of this Archean ocean. You see some tidal pools that may be boiling, there may be silicates there, and these carbonate structures, of course, is what the artist thought as being the earlier stromatolites. And of course, this is an artist rendering, and so we have to put some science in this art. And that has been done by many people. You see here some examples of fossil stromatolites, laminated structures, as you know. And we're going here. This is in the Pilbara. This is in California, where you see examples of very old stromatolites. The oldest stromatolites, and it's nice to touch on this maybe for a few minutes, very controversial in Greenland, and discovered by Martin van Kranendonk and Alan Nutman. You look then, of course, at this outcrop. You see in the dashed lines here some outlines of what presumably could be stromatolites. And, you know, with the first look, you can say, well, maybe this could be something. But of course, with anything we just mentioned, that going this far in time, you have to be careful and have to find multiple lines of evidence if possible. And so that is what Martin and Alan did. And one of the other things that can be used as evidence are these anomalies in the rare earth elements that you see here on the x-axis. If you see the anomalies at europium and yttrium, that may be indicative of certain conditions that are conducive of life. So these two lines of evidence and some other things are discussed in their 2016 paper. But then Abigail Allwood, who had prior to that the record of the oldest stromatolites in the fossil record, she visited the same location, and she wrote a paper saying, well, this is all perhaps not so straightforward. And one of the striking things that she found is you see here her sample with the two different phases of that sample that you see here. This looks like maybe like a stromatolite. You could be convinced perhaps that there are some laminated structures in here. But then if she looks at phase number 2, there is no lamination to be found. So the discussion is ongoing. Nutman and von Kranendonk say, well, she went to a different location. And I would just say stay tuned. But things like this obviously pop up every now and then. And I think it's very good and important to have a discussion in the literature and offline to see how can we find evidence, how can we convince ourselves that these were made by living structures. When you go a little bit closer, this is reminiscent of the structures where Abigail Allwood worked. You see these domal structures here. They are 3.45 billion years old, the Warrawoona and Marble Bar in the Pilbara. Again, not everybody would be convinced that these are actually biotic structures. We have to go a little bit later in time, maybe around 2.8 billion years or so, when we see more diversity in the structures, as probably most of you know. So the crux of my story is really, okay, if you look at the very famous the graph where you have on the y-axis the log scale of the CO2 in the atmosphere and time going from the beginning to present on the x-axis, you see an increase after the Great Oxygenation Event, and then you see the boring billion, and then you see another increase in oxygen. That's of course all tied to the geochemistry of the oceans and the rocks as well. You know the story, but really, if you look, and this is not on a linear scale, 30 to 40% of the time that we assume there was life on this planet, there was no oxygen, right? That's sort of the premise of my talk, the backdrop of my talk. So the question then is, how in the world did the ecosystems that evolved then, how did they operate? How did they function? Well, if you look at the composition of the ocean, again, from deep time more to more recent time, Holland said, well, first, of course, because of the volcanoes, we have lots of input of reduced iron with iron oceans. Then you have the Great Oxygenation Event, and then you get titration of that iron by the oxygen, and then you get oxic oceans, and then during some of these massive ice ages, you don't have photosynthesis, you don't have uptake of iron by biomass, and you can maybe get some iron oceans again, and then what we have today. Well, Canfield says, well, not so fast, and using sulfur isotopes, you of course constructed this boring billion where you have an hydrogen sulfide ocean that ends with an iron ocean, and then you get the oxygen. And Noah Planaski says, well, maybe there's flip-flopping going on. Whatever it was, before the Phanerozoic, we probably did not have large amounts of oxygen in the oceans. So if you then look at the work by Stucken, who has thought a lot about how these ecosystems would work, and in order to understand how a microbial ecosystem works, you need to know what are the electron donors and what are the electron acceptors, because microbes work through little redox reactions. And so the electron acceptors, basically what we respire with, there are different possibilities. In this from their 2017 paper, 4 billion years to present time, on a log scale, you see the concentration of a number of potential electron acceptors. And of course, as the ocean works today, you have photosynthesis using water as electron donor, making oxygen, and then oxygen is the electron acceptor, the reverse reaction using that organic carbon made by photosynthesis, and making CO2 and water. And here you have your oxygen cycle. And of course, today, that is how most ecosystems operate. So there are some other possibilities. Iron, of course, there's lots of iron there, and it has been proposed, and it probably did play a role, but the importance of that role can be questioned, because if you use iron 2 in photosynthesis, you make iron 3, which is completely, or is very difficult to dissolve, so not very accessible to organisms. And that's what we see in the BIFs, of course, the precipitation of iron 3 in the sedimentary record. So iron, very, very important, maybe not so much. Manganese is just present in very, very low concentrations, in nanomolar concentrations. So perhaps that was not so important. And also the chemistry of manganese is pretty complex. It's very reactive, and it may be very hard for manganese oxidizing organisms to compete for the substrate. And what we know from the manganese oxidizers today is that they exist in very special environments. Sulfur, of course, sulfate was not present early on because it was just not, there was not enough oxygen to make this, but there could have been a shorter sulfur cycle between hydrogen sulfide and elemental sulfur. So that's an important one perhaps to remember. But then, of course, nitrogen again used for biomass, so maybe not so important. But then there is this arsenic that was probably present in the Archean oceans as well. So that was sort of an idea to start looking at hydrogen. The last possibility was used probably for methanogenesis and for Luca-like organisms that used elemental sulfur and hydrogen for their metabolism. So not so important for perhaps for as electron donor for photosynthesis and in this respect. Okay, so that is sort of very, very quickly the backdrop of what is known and where the questions come from. So then you can go and hopefully find some analogs of these stromatolites that are currently alive. And this is an example of one of the Bahamian stromatolites where we did a lot of work. You see here at the subsurface, you see beautiful lamination. At the subsurface, you can see the green layer of cyanobacteria and a caramel layer at the surface there that forms a thin layer of this stromatolite. And Read et al. 2000 paper explains how the cycling of 3 different microbial mat types create 3 different fabrics in this coarse-grained stromatolite. And they're actually beautiful examples of this, as you can see here. So they have been studied to some extent, as well as the ones in Shark Bay. Also prime examples of living stromatolites. And then, of course, we do a lot of work in the intermediate between the stromatolites, a really lithified structure that is predominantly inorganic, and the more organic biofilms called microbial mats. This is an example of a hypersaline system from one of the lakes in the Bahamas, where you see some binding and trapping during storm events, and you see some precipitation in very thin layers of calcium carbonates. So what are these microbial mats and how do they make minerals is what I would like to spend the next 10 minutes on. So most of you know this, and please don't be offended if I review this very, very quickly, but microbial mats I would define as organosedimentary biofilms. So they are very organic, but they are very important in sedimentary processes. They trap sediments and they precipitate minerals and contribute to the sedimentary record. So some of them are completely inorganic. That's what you just saw in some of these examples in Shark Bay in the Bahamas. But typically, even those that resemble macroscopically, if you look at them as being completely inorganic or mainly consisting of calcium carbonate or other minerals, there is always quite a bit of exopolymeric substance, abbreviated EPS, in these. And what the EPS is, I will talk about in a few minutes. So they're sticky, they're biofilms, they bind and trap sediments, but the metabolic processes of the entire community can lead to changes in the saturation index, precipitating or dissolving minerals. There is a lamination of these microbial mats, which is mainly due to the light regime, and that light regime in turn has an effect on the geochemical gradients. Often the geochemical gradients are seen as the most important driver, but we know that most of the microbial processes actually take place in what is the oxic zone during daylight. It's where the cyanobacteria are near the surface, including methanogenesis has been reported there to be extremely high. Of course, through time, they've changed Earth's atmosphere through oxygenic photosynthesis, through nitrogen fixation, and as a byproduct of that nitrogen fixation, hydrogen production. And by the loss of hydrogen to outer space, the redox balance of our planet could have changed. These microbial mats have an incredible productivity, meaning carbon fixation potential. Within a millimeter of the mat, you can get as much photosynthesis, CO2 fixation, I should say, as you find in the rainforest from the floor to the top of the canopy. So really, really high compressed in just in a millimeter. And so as a result, you have, because these mats don't grow tens of meters per year, you have a very tight coupling of the photosynthesis and the respiration. So there's an enormous rate of element cycling going on, which makes them nice to investigate because there's actually something to study. Originally, we thought you could describe them by a limited number of functional groups of microbes, but the more we learn about the genetics, the more complicated this gets. And actually, we don't talk about species anymore, but about strains and even more importantly, genes and proteins that make these systems tick. It really is a mat, as you can see here. This is an example from the Orkney Islands. This is a top view, about 30 centimeters in diameter here. You can just pick up the mat and flap it over, just like you can do with your carpet at home. So they really give this coastal environment a lot of structural integrity. Okay, so let's review some of the ingredients of the calcium carbonate precipitation. How do you go from a soft mat to a lithified structure? Well, the first thing to look at is what the mat consists of, who is living there, and what are they doing. And one of the many ways to talk about that is to look at the short-term carbon cycle. The short-term carbon cycle, in contrast to the long-term one, is one that goes very fast. You go in that from CO2, the most oxidized form of carbon, to organic carbon, and including methane, through a reduction process that on our planet typically is driven by photosynthesis. It can also be driven by chemolithoautotrophy, like sulfide oxidation coupled to CO2 fixation. If any process that gives you a lot of energy, free energy, that free energy can be used to invest that in the Kelvin cycle or another CO2 fixing cycle to go from CO2 to your organic carbon. So in the current mats that we see today, oxygenic photosynthesis is the key player in transforming CO2 to organic carbon. And there are some other anoxygenic types of photosynthesis that will become more important when we talk about the Archean later on. The most commonly known is the one using hydrogen sulfide and making either elemental sulfur or sulfate as the product and fixing CO2 with the energy that becomes available. But the energy from light and reducing equivalents that this reaction and the ATP that this reaction generates. So then you go from organic carbon, that's what you and I did for lunch today. We respire that with oxygen to give us energy, right? The negative number of the delta G means that you get a lot of energy. The larger that number, the more energy you get. And so we engage in aerobic respiration or fermentation if you are a long distance runner. Microbes have a lot of other possibilities. They can respire nitrate, manganese, iron, sulfate, and even carbonate when they make methane, but the energy yield becomes smaller and smaller. Now, when you see the cycle and you see that photosynthesis is the major driver, you can already imagine that during a diel cycle, this top part of the cycle happens only in light, and the bottom part of the cycle, as long as this organic carbon reservoir is large enough, will go on 24/7. So there is sort of a decoupling of the top and the bottom of this cycle, which results in temporary larger reservoirs on either side of this cycle. And if the CO2 part of this cycle is enlarged, then you can get a loss to the atmosphere or to the lithosphere by making calcium carbonate in a very, very simplistic way. Okay, so how does it work? Well, let's give one example. Photosynthesis, oxygenic photosynthesis. You have CO2 and you have water as an electron donor. You make organic carbon and oxygen. I just told you that the rates are incredibly high, right? More than in the rainforest. So what happens is that you deplete your CO2. And so in a CO2 buffered system, which in our current atmosphere you will typically find, you will get a dissociation of your bicarbonate to CO2 and OH minus. That CO2 is immediately used in this green reaction and OH minus persists. So the blue reactions are chemical reactions that are actually pooled by the biogenic reaction. So you make OH minus, which means that if you have calcium and bicarbonate in this system, you can make calcium carbonate, which would make a proton. And you know that if you put acid on calcium carbonate on limestone, you go in the opposite direction. But if you have a way to remove this proton, which you can do through this OH minus, this is in this bottom reaction, then you can drive temporarily, you can go to a situation where you precipitate calcium carbonate during oxygenic photosynthesis in the light if you would isolate this process. So if you add up these reactions, you see that you have two bicarbonates of calcium to make some organic carbon, a calcium carbonate and oxygen. In other words, you produce one calcium carbonate per CO2 that you fix. Now you can play that with the same game on paper with a lot of different organisms, the key players in the microbial mat, cyanobacteria, the anoxygenic phototrophs, the sulfate reducers, and the methanogens. The light boxes indicate processes that help the precipitation of calcium carbonate by changing the saturation index in a favorable way, so you get precipitation of calcium carbonate. The darker boxes indicate processes that would do the opposite. They would create conditions that would dissolve the calcium carbonate, or they would lower the saturation index, in other words. Daytime and nighttime are different scenarios, as you see here. And you can just start making the balance of all these processes. And what we do in our lab, we go to the field and we can pretty much measure most of these in very small steps. At the micrometer scale, we can map the different geochemical processes and try to make a balance, a 24-hour balance of all of these at a different depths, and see how it plays out, and if you could have the potential to precipitate calcium carbonate based on the metabolic reactions of the entire community. It gets a little bit more complicated, though, if you look at the nuts and the bolts, and I'm not going into detail, but just as an example. So you have the CO2 fixation by the cyanobacteria. They may excrete some of the organic compounds that they produce during CO2 fixation. Heterotrophic organisms and fermenters break down the larger molecules to smaller molecules, which then are used by sulfate-reducing bacteria and methanogens, but mainly by sulfate-reducing bacteria in the marine microbial mats, that then ultimately change the saturation index. Now, it really depends on the products of this breakdown of the macromolecules, what the overall result will be. If you have sulfate reducers using formate or hydrogen, you get a huge change, a relatively large change in saturation index. If you have other compounds like lactate, actually you lower the saturation index. So in other words, it's not just important to know which organisms are there, but you need to know which genes are turned on and which proteins are turned on to see in more detail what the different processes are. And some people do that I collaborate with, and others. And we do transcriptomics to understand which genes are turned on during the 24-hour cycle, and that gives us a little bit more information. So there's more needed than just an overall idea of the community metabolism. Okay, so the second ingredient, exopolymeric substances, the slime. And you see here a picture of that modern marine stromatolites, SEM picture. And you see this biofilm draped over the surface of this stromatolite. And here are some confocal pictures Alan Deco took. You see in red here the precipitate at the surface of the stromatolite. You see some ooids here, and you see some autofluorescence of cyanobacteria in that. This other picture again, you see the ooid with some endolytic activity and some cyanobacteria here. And green is the EPS matrix. EPS matrix has a lot of roles. It's sort of the sweater that I'm wearing today, because outside it's minus 12 degrees. So I modify my environments. Organisms do the same thing. As a response to the environmental conditions, they make this slime, and the composition of that slime varies with the environmental conditions in a very simplistic way. And here you have the EPS matrix, polysaccharides, proteins, other stuff, and they have some chemical functional groups, like carboxyl groups, like phosphate groups, like sulfonate, sulfonic, great, so some type of sulfur functional groups, and amino groups, and some others even. And if these are deprotonated, if they have a negative charge, they can bind metal ions, including calcium. And that is very important in the next step of the calcium carbonate precipitation. So you have cyanobacteria and other microbes that make that EPS with those functional groups, this carboxyl group, for instance, that can bind calcium. So initially, the fresh EPS scavenges the calcium from the environment and actually prevents the precipitation of calcium carbonate. But at some point, it's like a sponge. If the sponge is full with water, calcium being the water, then the water runs through it. So when the saturation of this binding capacity is reached, you can get free calcium, and you can get the precipitation. The saturation index goes up, and you can get the potential of precipitation of calcium carbonate. You can also degrade this EPS. It's food, right? It's organic stuff. So some heterotrophic bacteria will degrade it, liberating the calcium and bicarbonate. You can also get, during diagenesis, some reorganization of the template. Tricia and Defarge has written on this, and create conditions also where the saturation index goes up, and you get a template for nucleation of calcium carbonate precipitation. And you can see that. So in the field, if you have a system, and one of our papers going back to the early 2000s, we described that so here you have an environmental SEM picture. You see the cyanobacteria the filament sticking out, and you see this honeycomb structure being the EPS. And if you expose this to conditions where you can get precipitation, we did a transplant experiment, long story, but we moved this soft mat to a part in the lake where there was precipitation going on, and we followed through time what happened. So like a month and a half to 2 months later, you see a lot of calcium carbonate precipitating in the matrix in this hypersaline system. And if you come back even later, it turns into a complete macritic layer at the surface of the mat. And the detail shows here that the EPS nucleates, the minerals nucleate, the calcium carbonate nucleate on the EPS matrix. And there are some reports in the literature actually of this EPS-like structure. This is from the Silurian in Morocco, published by Barbieri. You see that's very similar structure of the honeycomb EPS. So EPS plays an important role. Then there are some other maybe less known aspects of calcium carbonate precipitation. And one is the communication among microbes. There are a number of very small molecules that organisms use to talk to one another. And these are small and fairly inexpensive to synthesize, and they are used in a very clever way. So here you have a bacterium that for some reason starts to produce one of these compounds. This is an acylated homoserine lactone, an HL. It produces this, it releases it to the environment, it undergoes maybe modification in the environment, and another organism of the same species, typically we think, picks this up, and you see then a transport over the membrane and a complexation with a protein that results in binding with DNA and gene expression and a physiological change. So one organism releases this small molecule, another one picks it up, and you get a change in the physiology, in the metabolism of an organism. Now, if you look at you see here a chain length of, in this case, it's 6 carbons, but what we see in the real world, and this is for sulfate-reducing bacteria, we see different chain lengths. This is a type strain that you can buy off the shelf from a culture collection, and these are some of our isolates, and these are natural mat samples. We see sort of unusual in blue here chain lengths that are a little bit longer than what was typically known. And so when you look at those and you see the longer, so the higher the number, the longer the side chain is, the more stable these are under elevated pH conditions at very high pH, which is easily reached during photosynthesis. The short ones have a very short half-life. The long ones have a very long lifespan. So these sulfate reducers, they excrete different chain lengths of this molecule, so different types of this molecule. The other organism picks it up in the environment due to exposure to the high pH conditions. The short ones disappear and the longer ones persist. So if you have a ratio of 1 to 1 to 1 of 7 to 12 to 14, an organism just a few micrometers away, and has to happen in very close proximity, that's why it's called quorum sensing, may find a different ratio and can tell something about the pH of the environment. So let's skip this one. So again, the communication, turning on and sensing the environment is very critical. And here you see, again, an image of Ellen Deco in green, our sulfate-reducing bacteria that sit at the surface of this stromatolite where you find maximum oxygen concentration. So that was sort of surprising. It's a different story. We don't want to talk about that now, but we have some answers to how that may happen. Then another ingredient of lithification are viruses, and we learn a lot about that. Maurice Tucker has worked on this a bit, and we're doing some work on this. Viruses by themselves, since they have capsids of protein, they can bind calcium and calcify. And that happens. I just had kidney stones a week and a half ago, and that could have had a viral origin because the calcium binding happens on viruses. But viruses lyse organisms, and if they lyse organisms, they release nutrients, and so the photosynthesis rates can go up because the nutrients goes up. But by lysing organisms, if there's internal calcium, the calcium concentration locally can go up. Dead cells after viral infection can also help the strength of a biofilm. And so there are a lot of things. Again, it's a seminar by itself. EPS is very important because it can trap viruses. But EPS decay, so heterotrophic consumption, can actually enable viral infection because the protective EPS is gone. So it's a difficult story, but there are definitely multiple roles of viruses in the precipitation of calcium carbonate as well. All right, so you know a little bit about how this can work, and so now we go to a real-world example. Can we link what we find in deep time with something that we find today? So this last 10-15 minutes, I like to show you the combination of two field studies that may provide some insights. And I'm saying may because remember, in the beginning, I said we only have 5 or 6 pieces of this 1,000 pieces puzzle. And so you have to be really careful in how you interpret your observations. So here we see Western Australia, the Pilbara here, and the Tumbiana Formation, Fortescue Formation. Here are some images of David Flannery, who's done quite a bit of work there. Some of them look like really beautiful stromatolite from the top, from the side. And so we're going to look a little bit at that. Again, Western Australia here, 500 kilometers. Tumbiana here with different basins. The Hammersley group in blue, the Fortescue group in yellow, and some granite greenstone belts in pink. So here the blue and the yellow indicates sort of the parameters of what Tumbiana presumably was, a shallow lacustrine or lagoon type environment with lots of volcanic activity, not very deep. So with some microbialites that formed in that system. And this is another example of the Tumbiana Formation. You see really a wide variety of larger domes, some different shapes and different morphologies. You can see very small ones, large ones here, club-shaped ones there. So many examples of stromatolites found in the Tumbiana. And so Pascal Philippot went out there and did some drilling and crossed paths with me, and we did some work together. And the work that we did together was with Mary Sforna as the PhD student, Andrea Somoghi and Synchrotron Soleil, where you see an image of here, under construction still in this part, but somewhere here is a very long beamline called the nanoscopium. And the nanoscopium is currently the longest of the beamlines. There are many different ones. The synchrotron has synchronized particles in contrast to a cyclotron, where the particles are static. Here they are used in the various beamlines, and we are trying to combine 2 of these beamlines, a high energy one where we can see a lot of the metals, and a lower energy one where we can see the softer elements like sulfur, because the story that is going to follow now, in that we have an important role for arsenic that we can see in the nanoscopium, and for sulfur that we can see in the Lucia beamline. Okay, so you go with a thin section, very thin, 60 micrometers. You see it here. You put it in a beamline and you generate element maps, and the element maps in the nanoscopium can be with a resolution of nanometers, at the range of 10 nanometers or so. So that's more than a 10th millionth of an inch. Very, very, very fine scale, and so that is very helpful in interpreting what might have been in the past. If you can compare these maps, in this case you see the map of calcium, and the insert here is from the Sforna et al. Paper 2014, where you see high concentrations of calcium that overlap with high concentrations of arsenic. Evan Lepeau found that there were some organic globules, and these organic globules were actually affiliated with arsenic, whereas the sulfide and the iron were in pyrites associated, independent of the organic, or away, not in close proximity of the organic globules. So there was some evidence that perhaps this calcium carbonate structure that you see here in this element map could have some relation with arsenic and perhaps arsenic cycling. And that is what we proposed. But then you like to find some evidence for that. So the next step then is to have a lot of luck. Win the lottery or find a field site where you find the right conditions. So at some point, scouting microbial maps all over the globe, I was very lucky to connect with Maria Farias, and she took me to some pretty incredible places in the high Andes, and this is on the Argentinian side. We're close to 6,000 meters. We have lakes where you find actually microbialites growing. Pascal and I went off to a volcanic crater, Diamante, in the Argentinian side, and did some work there. I had to do measurements throughout the 24-hour cycle. Well, Pascal was sleeping in the morning, and it was minus 25, minus 27. And if you stick your hands in water, it gets really wickedly cold. But in the name of science, there you go. It was not the best field site because there was still oxygen there. But here in northern, so this was Diamante, so sort of here on the Argentinian side. But on the Chilean side, the Atacama Desert, again, the picture that I showed you earlier, that idea of the artist rendering of the Archean, keep it in mind. This is what the southern Atacama looks like. From this very shallow lake, you can count 35 active volcanoes, and you can find something really special. Again, Atacama Desert is here. The field site is in the southern part of it, and this lake is being fed by this red river. So the lake is here in the background. This stream that is feeding the lake is red. And why is it red? It's red because there is a purple sulfur bacterium called Ectothyroidospira that you can see in this layer. This is a microscopic image of it. It uses hydrogen sulfide to photosynthesize, and it doesn't make oxygen, but it makes sulfate or elemental sulfur. It also was found by Ron Ormond to use arsenite and make arsenate in photosynthesis. So it's an arsenotrophic photosynthesizer. The La Brava is at high altitude, UV 25 times higher than at sea level, very low humidity, a fraction of a percent. pH is on the alkaline side. And very interestingly, the diversity is almost 3/4 Uri-Archaeota, very, very unusual. Sulfide is high, arsenic is very high, and there is no chlorophyll A. But the photopigments for purple sulfur bacteria are found. Aragonite and calcite are inside the mat. Lithium is also very, very high, which I just was talking about earlier. Atacama Desert is the prime location for lithium mining. Bolivia has a lot of lithium, but it's low grade. The Atacama Desert has very high grade lithium. So we do measurements around the clock. This is in the summer, so not too cold. And we do different measurements. So measurements with microelectrodes. So this is in depth in millimeters. You see that there's no oxygen. This is the overlying water. This is the surface of the mat. There's never oxygen that reaches the surface because there's so much hydrogen sulfide in the stream that all the oxygen reacts and is removed. The sulfide is coming from the water column into the mat. There's a minimum of sulfide in about 1.5 millimeters due to the photosynthetic consumption by this Ectothyroidospira. At nighttime, when the light is gone, there is no consumption of sulfide, and this is your typical dissolution profile. Where you find photosynthesis, and a little bit below, you find a slight elevation of the pH to about 9, and so definitely sulfidic photosynthesis going on. This is the mat, as it shows again the pink layer. If you look at this small insert, this is the map, the synchrotron map of arsenic, and you see that there is arsenic present. We took 2-point measurements and did Zanus, and with that Zanus you can actually use arsenic 5 and arsenic 3 as standards, and you see in point 2 and then point 1 you see dual peaks, meaning that you have 2 oxidation states, and that is very, very important evidence. When you have 2 oxidation states, you know that you have the possibility of element cycling because you need 2 oxidation states to make redox reactions happen. So that was all very, very positive. Let's not talk about this. Is a sulfate reduction map, but we don't have to talk about that. So you can take pieces of this mat and make a homogenate, a slurry, incubate it for a while so everything goes away, and then you can give it a shot of arsenite, in arsenic-3 in this case, and do different types of incubation. Well, what this graph shows that over time, in about 20 hours time, you really need light to make this arsenite go away very quickly. If you give nitrate, it also goes, but much slower in the dark, and when you kill the bacteria, nothing happens. The similar experiment where you add a shot of arsenate, so this is the arsenic-5 compound, and you give some organic carbon, you see that over time this arsenate is being consumed and in the presence of organic carbon. And the scales are slightly different here, 300 and 600 micromolars. Basically, what you can see in the next slide is that the rates in blue are the arsenic rates. We did similar experiments using the sulfur cycling. You see that the arsenite utilization is almost the same rate as the arsenate. So again, very, very fast cycling. On sulfur, the potential of sulfide oxidation by phototrophs is actually much greater than that of the respiration side. And we think that it is actually suppressed in this arsenic system. Both happen, but suppressed. Next line of evidence is that we see different arsenic-related genes. That the three of them are mapped here, and the breakdown is not so important, the organisms that contain them. But basically, if you know which enzymes are there, you can make the arsenic cycle happen, because all the enzymes, all the genes that make the enzymes for the different enzymes that cycle arsenic are present in the sample. And so you can go from arsenic-3 to arsenic-5 in the light, predominantly making arsenic-5 that you can then breathe with organic carbon to go back to arsenite. So complete arsenic cycle. We found the geochemical evidence, we found the genes, and we found the two oxidation states. So plenty of evidence to make us believe that arsenic cycling is important there. Well, why is this arsenic cycling so important? Well, if you look at the reactions on the respiratory side, so on the breathing side, if you breathe hydrogen with sulfate, or you do it with arsenic, there is a much greater, the bigger this number is, the bigger the effect on the saturation index. In all cases, the red reactions, the arsenic supported reactions, give a much higher negative number, meaning have a bigger impact on the saturation index. So in other words, make the calcium carbonate precipitation potential much greater. So then we can come back to that short-term carbon cycle, and now we add in arsenic on the respiratory side and also on the photosynthetic side. And again, looking at these numbers, you see that arsenic's actually, the respiration with arsenic is slightly more favorable than the use of sulfur. So it makes sense from an evolutionary point of view that arsenic cycling was more favorable and evolved earlier, or had a potential to be more important in ecosystems than the sulfur cycle did. So before the GOE, no oxygenic photosynthesis, or it started to come, no aerobic respiration. Nitrogen probably was very limiting and used for biomass. Manganese just not present enough, maybe some iron cycling, but arsenic was really one of the dominant possibilities for respiratory processes. And arsenite was definitely one that could have sustained the photosynthesis by purple sulfur bacteria or organisms that were like those. So then, coming back to the summarizing slide, volcanism, weathering may have delivered arsenic-3 and hydrogen sulfide, hydrothermal processes even so. And you get an active sulfur cycle, perhaps a short one, maybe only from hydrogen sulfide to sulfur back to hydrogen sulfide. And in red, from arsenite through photosynthesis to arsenate, and respiration using arsenate back to arsenite. And both of these element cycles would have an impact on the production of calcium carbonate in the earlier stromatolites. So close to La Brava is another lake called Tebenkiche, where you have a lot of these structures. Actually, they're gypsum and not carbonate, but anyway, so you can envision this perhaps on early Earth in the intertidal zone, the production of these structures on a very large scale. One last thing to mention is that, of course, the Martian rover is running around now, and I think Karim may be online, and he's involved with these missions. The current rover, the latest rover, the 2020 mission, has a pixel instrument, planetary instrument for X-ray lithochemistry, which uses basically the same technique, but does not have the same resolution as what we have in synchrotron Soleil. So who knows? Maybe one day we find arsenic cycling, or evidence of arsenic cycling in the past on the Martian surface. That brings me to the end of my story. We had covered a lot. I hope there are some questions. Obviously, this is the work of many, many people. You see the long list of some of the collaborators here and my funding sources as well. And I thank you for your attention.
Yeah, so thank you very much, Peter. It was really interesting. Is there any question in case you can maybe raise your hand, or just yeah, if you want to ask any sort of question. Yeah, I do have a really small one. Maybe it's something I've missed, but yeah. So you said that in the sort of living mat that you were studying in the lake in Chile, so you've spotted arsenic 5 and arsenic 3. That was it was one of the proof of your that there was arsenic cycling, so it was maybe used as in the respiratory cycle. Can you make such kind of measurement on fossil microbials and find such kind of isotope presence, or?
So this is elemental presence. In principle, yes, but of course, and the deeper you go in time, in geologic time, the more difficult this gets. You know, these rocks get cooked, and you get alteration, and so it is difficult to say. If the further you go in the rock record, the more difficult is it to say that this was actually present at the time that the microbial was formed. But it gives us, yes, it gives us a possibility to better understand the modern systems, and then maybe we shouldn't go all the way back to the Archean, but maybe we should look at some systems that are a little bit more recent, like for instance, Emmanuel Benin in Dijon has worked on the structures in Great Salt Lake and Rapouillot in Bordeaux, and I've had the pleasure to work with them there. And so those are systems that are much younger, and maybe you try to take that step first and then go deeper and deeper in time. One of the things that I didn't show today, but what I'm doing in my lab is I have worked with the engineer of Elvin, the submersible of Woods Hole, who has built a system for me where I can mimic diagenesis. I can, in a couple of months' time, I can mimic 3,000–4,000 years of diagenesis. Of course, not perfect, but by high temperature and high pressure, I can put an entire core in a device and do some measurements before and after, and that is something that probably has a potential to understand that first step of alteration of the elemental composition. It happens, obviously, there's no question about that, and it's again only one of the pieces of the puzzle. We need multiple lines of evidence, but it could be a promising one. Who knows? But a good question.
Okay.
The answer is a little wishy-washy, but yeah.
Okay. And maybe I have a second one, a very small one on chrome sensing. So just you said that basically by producing different size of molecule, the protein were kind of able to measure the pH of the environment. Did you see any change of not physiology, but change of behavior in the bacteria due to this pH sensing, I would say?
That's a very good question, and I wish I could get that. I'll very quickly fly through this. I can go back to the one I didn't show. So actually, what we see is that's the answer of perhaps why you can see these very high rates of sulfate reduction, perhaps I said, in the surface of the mat. So you have the cyanobacteria that produce organic carbon that the sulfate reducers need, but they also make oxygen that the sulfate reducers maybe don't like that much. So they say, whoa, there's something going on. they say maybe the organic carbon is there. Now putting brains in the bacteria, right? This is all very hypothetical. So they say there's organic carbon there, let's ramp up our metabolisms. But they also, the same compounds that they make, we have some preliminary evidence that these quorum sensing compounds, some of the very long ones, actually affect the sulfide oxidizing bacteria. Sulfide oxidizing bacteria use actually the oxygen, my cursor is jumping a little bit, the oxygen and the sulfide that is produced by the sulfate reducers. That sulfide is actually toxic to the cyanobacteria. So if you have three groups of organisms working together, the cyanobacteria making the organic carbon and the oxygen, the sulfate reducers using the organic carbon and making hydrogen sulfide, the sulfide oxidizers using the oxygen and the sulfide, everybody's happy and everybody can work at its maximum potential rate. And that's what we can actually measure in these microbial mat systems and microbial systems, that they work at incredibly high rates, close to the maximum that we know from lab experiments with individual cells. We do not know if the quorum sensing compounds of the sulfate reducers are picked up by the cyanobacteria, but we do know that the quorum sensing compounds made by the sulfate reducers are picked up by the sulfide oxidizers. So again, there may be a potential here. The problem is that it's not very easy to measure all these compounds at the same time, and so it's the top of the iceberg, I would say.
Yeah. Okay. Yeah. Thank you. So Maria has a question. So maybe Maria, you want to ask the question directly?
Maybe Geoffroy, Karim had a question before.
So maybe Karim, then Maria.
Yeah, okay, no problem. Just one question, Peter. Yeah, for the arsenic speciation, have you tried exapts and to understand where exactly arsenic is and whether this is within the structure in the carbonates? I mean, I guess arsenate would be more like a phosphate group, but arsenite would be something else. Or is it related to the organics? And because I guess that will also control the fate of this of arsenic along diagenesis.
Yeah. So that's a really excellent question, and so we're trying to figure this out. And initially there seemed to be somewhat of a trend that if you would have high arsenic environments, some of the arsenic would end up in the carbonates. But if you have a highly reduced environment, it can also be incorporated in the pyrites. And then, of course, it can still be part of the organics as well. But we see some of it definitely in the carbonates. We see in with the synchrotron. We can see beautiful maps of veils of EPS where the arsenic is sequestered. So in the modern mats, that's definitely a sequestration mechanism, like the calcium, of course in a slightly different way. It must be, but we see that first, and then it depends on the degradation probably of the EPS and the amount of arsenic versus calcium versus other elements where it ends up in the end. But it's a very good question. The puzzle is a little bit more complicated than we initially thought, and we're still trying to work on this. But we see it in carbonates as well as in pyrites. That's the short answer.
So maybe Maria, then Lucien.
Yeah. So I was asking myself, did you in your recent maths, did you have the possibility to look at any intracellular arsenic and especially its localization?
So that, again, it's a very, very good point. We have not done that in the Atacama mats. Of course, there are, in addition to the oxidation reduction reactions utilizing arsenite, arsenate, detoxification mechanisms that have been studied in quite great detail. There is metallation going on of arsenic, making metal arsenites, and volatilization of this. I think there are some reports, there are some ideas that perhaps the arsenic is found in internal calcium carbonates that some of the microbes produce. It's not our work, but I've seen some presentations on that. So I think that it's a very good question. I think my techniques wouldn't really lend them to investigate that, but would be fabulous to do a nanosims experiment and do some mapping or something like that. I think it's a very good point. Yeah. Yeah.
So yeah, Lucian, if you want to ask a question.
Yes, Peter, thank you very much. Wonderful talk. I was wondering if you can comment during the anoxygenic times, there was arsenic detoxification or only respiration?
Again, a very good point. And then you get to the difficult question of when did certain pathways arise. Wolfgang Nietzsche in France has done some work on this, and actually some of the ideas that have I think come to the foreground is that there were detoxification mechanisms. The detoxification mechanisms may be again older than the cycling mechanisms that we know today. Of course, one of the issues we're always dealing with going into deep time, if you the tree will no longer, I mean, it's too simplified now, but if you would create the image of a phylogenetic tree, there could be a lot of dead branches on this tree, right? Who is to say that that there were not organisms that are no longer present and have not evolved any further than what we find in today's microbial mats and microbialites. So it's always hard to say what exactly in very deep time, the beginning of life, what kind of enzymatic systems, what kind of metabolisms there were. So even if it is true that the detoxification mechanisms are older than the redox cycling mechanisms, it's not to say that perhaps some other ones have existed prior that we no longer are aware of.
I asked because I work on a Shebanella that possesses both respiration and detoxification genes, and it's not the only examples. There are other examples.
Sure. No, and that makes sense in definitely contemporary environments. That would make a lot of sense. Yeah.
Thank you.
So, is anyone else question? I don't see any raised hand. Okay, so well, if we don't have any more question, I think we're just on time for the end of the seminar itself. And then I will say thank you very much, Peter, again. It's been really a pleasure to organize this seminar with you, and it's been, I think, really interesting. And thank you all for attending today. And we will see you in maybe a few weeks for the next talk. We will communicate about that as soon as possible. Yes, Juliette, maybe? Yeah, in actually 3 weeks from now. So you will receive really soon an email for the next seminar. Yeah. So stay tuned.
Great. Yes. Well, my pleasure, and good luck and stay safe and healthy, everybody.
Yeah. Thank you. Thank you very much. Have a nice day. Have a nice day.